In this video, I'm going to be talking about the synapses of chromosomes, specifically the synapses of chromosomes in two separate individuals and a crossover is an event between the J and K genes and between two of their chromosomes. So for individual 1, we can see that their chromosomes right here. We can automatically tell that there would be a chromosomal aberration in the middle here across the centromere because these letters are inverted from each other when you compare them, meaning this is a paracentric inversion. In order to, and it helps to draw a diagram like this in order to determine the chromosomes that are produced in meiosis from the crossing. So what I did is I drew out the chromosomes, one chromosome represented by one line, and I traced it out with a different color to represent different chromosomes. So for this light blue right here, I traced it out, and as I crossed a gene represented by the letters, I wrote it down and recorded it. So for this blue, I got M, I, J, K, L, H, G. As you can see, only 2 of the genes are interacting in this crossover event. So this gene was not interfered with in the crossover. And I also record the centromere represented by these 2 black dots. For red, I traced it, and this one would be involved in the crossover. So I traced it, M, I, J, and then crossover K, L, M. And now for the green, M, L, K, J, I, H, G. And for the last one, the yellow, G, H, L, K, crossover, J, H, G. So now to determine the viability of each of these chromosomes, we're going to see if they have a duplicate or they have more than one centromere. So after looking at that, we can tell that strand 1 and strand 3 are viable. But strand 2 has a duplication of the M genes, strand 4 has a duplication of the HG genes. So we have a total of 2 viable chromosomes in individual 1. For individual 2, we can tell that there are no chromosomal aberrations because it was exactly the same. So we can draw it like this, and as you see, through the same methods of individual 1, we get 4 chromosomes that look exactly the same. And they are all viable because they don't have duplicates and they only have 1 centromere. So we have a total of 4 viable chromosomes for individual 2. So determining which individual will be more fit, we would have to say individual 2 would be more fit because it has more viable chromosomes to pass on to the next generations and to survive the next generations. Individual 1 only has 2 viable chromosomes.